Hello, my name is Jeff Martin, and I'm here with another Jeff Shumway from Integris. What is Integris doing, Jeff?
Well, Integris at its core is actually a material science expertise company, and it traditionally has supported the semiconductor industry. But within the last several years, we've developed a portfolio of technologies that are directly applicable to life sciences.
Such as,
filtration. So in 2001, Millipore actually spun off their filtration portfolio called Microlis that supported the semiconductor industry. That became a piece of Integris. ATMI, a little piece of that, became part of Integris through acquisitions. And a company called Flex Concepts, based in Logan, Utah, that specialized in single-use manufacture, was acquired by Integris to kind of build up that life sciences portfolio. So the filtration heritage and the legacy of knowledge within this space has always been there, but we're a relatively new player to the life sciences space.
Yeah, so when you say new, like how new? When did you guys kind of get into this?
Within the last 7 to 10 years.
Really, so not brand spanking new.
Not brand spanking new, right? Yeah. And our portfolio is pretty specialized because of that material science expertise. When you think about particles, for example, particles are a huge topic of conversation within cell therapy manufacture, and controlling the risk around those has been something I've been focusing on over the last couple of years. And when we think about particle control and particle risk mitigation, for us in life sciences, it's orders of magnitude more important in the semiconductor space.
Yeah, I guess you're right.
Yeah, because you think about the costs associated. Now, we talk about effects on patients, which are, it's a different dynamic, but the level of scrutiny in the semiconductor world is much, much higher than it would be in our space.
Right. So I guess the question was, what got you guys interested in coming into cell therapy?
Well, a lot of it is fit for purpose. So we have this film that it's a very, very high purity film that will withstand temperatures of minus 196 C and still be flexible and pliable.
Interesting.
That same technology can be made into the porting material, so it's a single material of construction for the whole single-use bag chamber. And that provides some interesting advantages. Number one, chemical compatibility. This film will tolerate 100% DMSO without breaking down or fracturing, which is a common material used for cryopreservation. Number two, it's essentially particle-free. The material is such high quality, it could be used to package a silicon wafer as much as it could be used to contain a cell therapy drug product. And number three, the tolerance in those low temperatures gives it these wonderful performance attributes that make it directly relevant for this industry. And what we've done as an organization is we're pretty agnostic when it comes to the componentry. So by partnering with select partners like, for example, DuPont and their new low temperature TPE tubing Livio, we've got an agreement with them to be able to put that onto our chambers, and then suppliers like CPC with their low temperature connection technology, we can put together a very well fit for purpose technology. Additionally, we have these filtration capabilities, these legacy, and only recently have we launched our PharmSteri product offering, which is a series of sterilizing grade polyether sulfone membranes of different layering strategies.
It's a mouthful.
that is, at the same level of quality and performance as some of the bigger players in the industry. And because we're a newer player, we have a little bit of an advantage in terms of our costing structure.
Sure. And so what are some of the things that you guys mainly, supply? You mentioned filtration systems. What else is there? Because I feel like you probably have a lot of capabilities with the technology that you're working with there.
Yeah, so single-use fluid management is one of our core competencies, as is filtration. But in addition, we actually have a whole group that is in a specialty chemistry area. So a lot of the base technologies for synthetic nucleotide synthesis, we have technologies around that. And we're constantly evolving.
That's very different from, plastics.
Exactly. Well, it's polymers, right? It's just biopolymers. Versus,
I guess at a certain sense, we're all polymers, right?
Exactly. So we're constantly identifying new places where we can benefit this industry.
Yeah.
Leveraging adjacent technologies, right?
Yeah.
That come from a very different world.
How common is that? Coming from superconductors to biotech, who else is doing that? I mean, it's got to be,
It's interesting if you look at even Millipore Sigma, which is, I spent a few years there. Merck KGaA in Germany has 3 different divisions. One is healthcare, where they're making drugs. One is the life science technologies, where the Millipore Sigma piece fits. And the third one is actually microelectronics, so semiconductor tech.
Yeah.
So it's not that novel. Has it been leveraged well? Well, when you get to organizations of certain sizes,
Sure.
it's really hard
It's
to
rigid.
access those capabilities even
Yeah.
if you have them.
Yeah.
And I'll give you an example. There's a detector technology that looks at concentrations based on refractive index changes. That's
Okay.
used very regularly over in the semiconductor industry. These are made of high purity, low extractable materials with very, very robust chemical compatibilities.
Right.
When you think about purifying a polysaccharide-based vaccine product, you can't look at those with a traditional UV,
Right.
but these refractive index monitors actually
That's
have
interesting.
a place to play.
Right.
Likewise, particle detection. Particle detection is critical for semiconductor manufacture.
Right.
We can apply those same particle detectors to looking at and qualifying materials that might come off of our filters or might be present in our bags, and we can optimize our processes to make sure that there aren't any.
Right. So how do you guys go about discovering a use case? Because it seems like you're just mentioning these things like you just kind of knew it, but how do you figure out like, oh, we can use this technology that we have to do that step in this process?
Well, a lot of it is identifying the pain points in the industry. Right?
Yeah.
One of the biggest pain points, go back and look at the 483s that were written against Novartis when their cell therapy Kymriah came out, right?
Okay.
It was all about particles.
Interesting.
And they discovered that those particles weren't coming from their process at all. They were coming from the bags that they were putting their cell therapy into.
Okay.
Now, the positive thing there is that they were catching these issues.
Yeah, that's true.
The negative is that they were there in the first place.
Mm-hmm.
And if you read into those, you'll see that these were common contaminants of things like cellulose and wood and metal fibers, which are, if you're pulling film off a roll
Sure.
and making a bag out of it, those
You
are
get some
normal
dust
things.
around sometimes.
Exactly. And you take those approaches and you say, well, what can we do to improve on that? Well, if you're shipping a silicon wafer, you need to make sure not only that the bag is clean, but it's also static free. You reduce the static load, you reduce the ability of that film to attract particles to it, and now you've taken one step further to reduce the potential for risk.
Right.
Then when you manufacture, if you manufacture in a Class 7 environment, which is the same environment that our customers are going to manufacture their cell therapies in, you're solving a pain point that already exists.
So this is a little bit off topic, but I've seen some pictures of like semiconductor under a microscope, and a flake of dust is like the Empire State Building being laid down in New York City. It's
Yes.
like that. So
It's that dramatic.
it's that insane.
Yeah.
So yeah, I mean, I guess you wouldn't think that those kinds of standards would be required in cell therapies, but it's good that there's companies like this who are out there that are capable of meeting that standard.
Yeah, and it's interesting. We look at the ISO classifications, right? For a little while, we were manufacturing in an ISO Class 5 environment because it was a legacy semiconductor facility. And what we found was a lot of that classification, it's how you validate that space.
Sure.
The cleaner you can make the incoming raw materials, the higher standards you can put on those, and then monitoring and measuring your environment, not necessarily to qualify to ISO 5 or ISO 4 or some of those really more intense
insane
standards,
ones,
but leveraging the practices that the organization is using on that other side and the same quality systems.
And it's good to know that we probably don't need to go that high. It's a little bit overkill, but we can if we needed to, which means just crank it down a little bit, and we know that we're good to go at these levels.
Well, yeah, and you think about attributes like shelf life. I mean, a lot of the shelf life determination that we use in our industry is based on validation. And those validations are run maybe to 3 years. And the reason for that is cost.
Right.
The longer you run one of those studies, the more it accrues cost. We could qualify a facility down to, 0 particles per square meter, right?
Right.
would that be
Where's
cost
the
effective?
added benefit?
Yeah,
Right.
where's the added benefit? And even in the excipient industry, which is these are the things you're putting into that final drug product that aren't the drug.
Sure.
So they've got to be strictly monitored.
Yeah.
There's a terminology called TUPS,
Okay.
technically unavoidable process particulates. And what that industry has done is not only is they said, okay, look, we know that zero particles is unachievable. We're trying to get there, but part of making sucrose, you're
Right.
going to end up with some
Where's that tolerance
carbon
level?
particles in there.
Right.
As long as we know what they are, and as long as we control the limit and try and make them as low as
Sure.
possible,
Yeah.
that's one attribute. The other is, coming from the electronics industry, there's the concept of zero defect.
Okay.
Zero defect came out in the 60s, and the whole concept there was they knew they weren't going to get to zero defects, but by striving to try and target that, they ended up reducing their goal by over 50% in terms of the defect rate they were seeing. And one of the questions we've been asking internally at Integris is, yeah, maybe we're not going to get to zero particles, but can we lower the risk to the point that
Right.
it's not an issue anymore?
Right.
Yeah.
So what kinds of companies do you work with the most?
So we have pretty much two classifications of customer base and partners. One is the drug manufacturers themselves. So cell therapy manufacturers, lentivirus manufacturers, specifically in the novel modalities, because
Cool.
what's interesting about our premier film technology, Aramis, is not only does it have these really interesting compatibility and fit for purpose attributes, it's also really, really low non-specific absorption to some of these materials.
Okay.
So like lipids won't stick to it, and
Sure.
nor will viral particulates, nor will cell therapies. And in that space, we're doing fairly well, but we're also seeing a lot of traction in the suppliers of critical raw
Yeah.
materials.
Yeah.
Because they're looking
makes
for
sense.
solutions that can plug and play
Yep.
with similar materials of contact for high value components, so like cytokines for
Right.
cell therapies, cryopreservation medias, custom media formulations, where you want to make sure that material ends
Everything
up
is what
unadulterated.
you know what it is.
Exactly.
Yeah. That's it. Okay. All right. Well, it's good talking to you, Jeff.
Good talking with you.
Yeah.